So I'm also a fan of Star Trek. And, do you ever notice when the Klingons are about to attack? They get everything ready, the Klingons attack, and a few minutes later, everybody's falling all over the place with head injuries and all sorts of traumas, all of which could have been prevented. The reason they're not prevented is because there's a radiation physicist in charge. If there was a physician in charge, he would have said, put on your safety belts. So dose volume histograms, they're pretty damn good. Anyway, so I'm delighted to be involved in this physician versus physicist collaboration. So what we will be talking about is the partnership that exists and how we can elaborate upon that. So as a measure, what we can achieve with collaboration, this is a recent publication from Radiotherapy and Oncology based on a trial that Cancer Trials Ireland took part in. It's about DCIS. It's the biggest ongoing trial where we're awaiting the overall results. And as a small little country, we managed to get 2 authors from Ireland onto that paper because of collaboration through Cancer Trials Ireland being associated with other international organizations. So what type of trials do we involve ourselves in the St. Luke's Radiation Oncology Network and in Cancer Trials Ireland, the radiation oncology community in general? First of all, R&D. So not just pure research, but development. So when IMRT came out, Ireland lagged behind its implementation, and we found a very efficient way of bringing it into the mainstream was to create a clinical trial where the rules, the SOPs were all worked out. We cut and pasted what other countries had already done, and we set up a very successful ICORG 0817 clinical trial, which not only succeeded in its endpoint of implementing it for prostate, but meant that we used it for a whole load of different disease sites. So that's the R&D as opposed to just the R model. We're also involved a lot in national cooperative randomized trials. We had a spinal cord study which was presented at a plenary session at Astro based purely on cooperation within the country. And then the thing that we're most interested really are the international cooperative academic trials like the one I just mentioned. And one of the very key external collaborators for us is the Radiation Therapy Oncology Group, which has now been merged with the NSABP to create NRG, or NRG, nice acronym. And that's our top priority, but there are a lot of barriers to putting patients onto trials like that. Finance, the fact that sometimes the technology presents real challenges. And also approval. There are a lot of really attractive UK trials, but the organisation for approving and getting those trials underway in the UK will always prioritise Sheffield versus Ireland, and we have a real problem with that. And the EURTC, which is the European organisation, is very drug-oriented and not sufficiently radiation-oriented from my perspective. And of course, if you get into international cooperative industry trials, they're very well funded, but the huge emphasis is on drugs, and there are plenty of other people investigating drugs. So we do that if the drug is particularly interesting from a radiation perspective, if it's a radiosensitizer, but we're not really that enthused about drug trials. So we're focused instead on the technology itself, and you only have to look at the impact of technology on our life in general to appreciate how it has affected radiation oncology. So this phone cost about 650 quid when they came out early on. The only thing they were useful for was driving along with the phone in your hand with the window down pretending to be talking to somebody. They were actually very unfunctional, and now look what they've become. And so technology has had the same type of influence as we saw so eloquently in today's keynote address. And so in the late 80s, early 90s, that technology started to have a real impact. You will be familiar with computer-aided design, and that's really what drove the concept that radiation, which was delivered with basically a 2-dimensional technique, should really be delivered with a 3-dimensional technique of design and of delivery. And that has continued to evolve from early experimental clinical trials and technological innovations to mainstream innovation. So this is an example of a patient treated with a parotid cancer at the Beacon Hospital, actually with intensity modulated radiotherapy. And here are the targets here. And you can see the exquisite conformality. You're able to really confine it to the place you want to confine the dose to, spare the opposite parotid. And most importantly, if you go to the bother of contouring things like the brainstem, the upper lip, the mandible, you're able to put them in as organs at risk and optimize the dose distribution to spare them. So that's the kind of basic template of technology we're using. You've also heard earlier about motion management and how that's essential for stereotactic for lung tumors. We have some stereotactic trials underway. Dr. Alina Mihai up there has been a principal investigator for some of them, one international trial, and we have a huge, big international trial coming downstream, which she will again be PI for. And you heard Cora's work on stereo. What's our current translational research? The point I want to make here about the value of translational research linked up to clinical trials is that in the past, to a fault, you had really very brilliant laboratory scientists trying to gather human specimens from their colleagues and getting them by the grace of God, as opposed to a standard operating procedure, and never really having a perfect handle on the clinical data that belonged to that laboratory specimen. And then when they did their analysis, not having perfect systems for collecting the outcome data, be it survival or toxicity. When you attach laboratory research onto a clinical trial, your lab research, of course, has to be conducted according to a very meticulous protocol, which is predetermined in the trial documentation, and any change from that has to be done by a process of approval. And the data that's collected is collected according to the technique of ICH-GCP, which is a pain in the proverbial, but it does mean that everything is meticulously recorded and all the source documents are available, and it's all collected in a database. So really what you're able to do then is have quality correlated with quality. And that's really the real gold mine that we have when we do clinical trials and attach on translational activity. So we have some ongoing. Professor Pennington in the Conway, we're doing proteomic analysis of 2 prostate trials. We're extending that to some of our lung stereo trials. Professor Ling at DIT, Raman spectroscopy and normal tissue toxicity. Some very interesting data on one of our prostate trials. We're going to do it on lung trials, and we've also got some on non-trial head and neck data. Professor Brian Hennessy, in collaboration with Brian O'Neill at Luke's. Brian Hennessy is in Beaumont, P13 kinase inhibition, and we're gathering human samples for his laboratory analysis on that particular trial. So in terms of what opportunities do I see that just jump out at you? CT scanning of cells, we've heard about that this morning. This is the moron's way of describing some very eloquent work that was described already this afternoon. Can we identify radiotherapy-related changes, both in normal cells and malignant? And does that correlate with the overall outcome and with the other translational analyses that we're doing? So here's the Brian Hennessy, Brian O'Neill trial. It's the TRILARC study. It's a randomized trial of 3D versus IMRT for rectal cancer. And it's done preoperatively. And the patients are having a lot of analyses currently, whole blood, tumor analysis, circulating DNA and circulating tumor cells. And then there are 3 points in time when tumor specimens are obtained. So to me, that seems to be a fantastic template to which you could attach the UCD physics stuff we've seen already today, which I'm just going to call augmented microscopic analysis. I think that's a phenomenal opportunity. The trial is there. This trial allows us to modify it. This trial is going to be a trial that will extend to just keep on going, asking new clinical questions with the same background of gathering data and gathering specimens. So it's going to be, I think of it as the gift that will keep on giving. So you've already seen this, I won't repeat it. That's what I refer to as augmented microscopy and this. And we've also heard today about the issue about adaptive radiotherapy. An MR-Linac costs, I think, 6 to 7 million, but you don't have to have an MR-Linac to tell you that you've got a problem. Here is a patient with 2 completely different rectums and completely different bladders. We can see this today on our treatment planning CT scans. Seeing it is not the problem. The problem is being able to adapt to it in terms of your work organization capability, your analytic capability, and to be able to come up with a plan on the fly. That in a busy radiotherapy department is a formidable challenge, which is going to take a lot of rethinking about who does what in the radiotherapy department, physicians letting go of control of certain key things. Like I sign plans where I look at a dose volume histogram and as long as the thing comes below the dose volume histogram, why am I looking at it? The computer already knows that the doses are planned below the thresholds for safety. So why do I bother looking at it? Because I do. And I spend a lot of time doing it all the time. So we need to eliminate all that rubbish and just decide for the majority of the common cancers we treat, the machine decides, the machine gets on and treats it. With the therapists looking after the machine, of course, and the physicists. Brendan has a big interest in flash therapy, which I don't understand very well, but certainly very interesting. And the point that to be made about that is we have a research LINAC at St. Luke's Hospital, which is capable of delivering this type of ultra-high dose rate. So just realize that a LINAC costs a couple of million to begin with. It's there in St. Luke's. It's not being used for patients. So there's an enormous facility there for people to hook into in collaboration with Brendan. So in summary, we really have got established cooperations. I think we have a fantastic opportunity to expand this to include UCD Physics and, of course, Vincent's and Beacon, and align innovative science with existing clinical trials. Thank you.